So, when we say a transcription factor binds to the upstream region of a promoter of a gene, it either has a capacity to enhance transcription or repress the transcription of that particular gene. So, to check the binding of transcription factor to the upstream region, we basically clone the transcription binding site of transcription factor in a vector and then perform Lewis-Gray's assay. In order to do so, we need to design primers for cloning the binding region of transcription factor, and which basically includes four steps. So I'm gonna tell each one of it, but I'm gonna show one slide which shows all of these steps and the tools which we will be using, and which other tools which you guys can also use. So starting off the steps, first step is basically you need to retrieve the sequence of upstream region of that particular gene. And then second step is you need to check the transcription factors which binds to the upstream region. It could be one transcription factor or the particular transcription you want to look for, or it could be there are like many multiple transcription factors can bind to the upstream region. So you can do both the thing. You can check for particular transcription factor, or you could have multiple transcription factors. And third is the study of the vector which you are going to use for cloning and the restriction sites present in that vector. And fourth is designing of primers. So in order to do so, you can use multiple tools which I have shown at the right side of the slide. We will be using a gene called VGF, okay, and I'm going to show how to retrieve the sequence and look for the transcription factor binding sites in the upstream region of that particular gene. So to retrieve a sequence, you can use different databases, for example, NCBI and Ensemble. Here I'm going to show an example of a gene called VGFA, which I have retrieved from the database NCBI. You go into the NCBI site and you search for you type your gene name in the search dialog box, and the result you'll get. If you scroll down, you'll see a option of RefSec gene. You click on that option and you'll get the sequence of the whole gene, and it will on the rightmost corner of that result you will see an option of whole sequence or selected region. So for example, here this gene starts from position of 4996 and it ends at 21272. So from it's starting from 4996. So we want to see a region which is upstream to it. We are going to use 1 kb upstream, that is 1000 base pair upstream. So what you will do is subtract 1000 from the starting position. So and you will select that region only. So you'll update your view and you'll get the sequence which is upstream to that particular gene in the FASTA format. Once I've retrieved the sequence, I want to know the transcription factors that could bind to to that upstream region. So we are going to use a database called PromoLGen, which has multiple factors in it. So we are going to select species to human factor, and factors we can select factor option. We can select one transcription factor, or we can also select multiple factors at a time. So supposedly I've selected HIF1 and I'm going to then I'm going to put the sequence I've retrieved from NCBI in the FASTA format, like I've shown it in this slide. Here we got only one binding site where HIF1 binds to the upstream region of VGFA. But it is a possibility that a transcription factor has multiple binding sites in the upstream region of any gene. So what we do, if we have multiple binding sites, then we prefer that site where dissimilarity ratio is very less. Okay. As I've already told you that this software gives you option of selecting different multiple transcription factors at a time. I have shown this slide just to let you know that multiple transcription factors can be selected at a time, and you can see the binding sites of these transcription factor, uh, and you can also see like the distance between these binding sites or if they are overlapping or not. Once we have find the sequence at which this transcription factor is going to bind, then we have to clone this sequence into a vector. We are using PGL3 basic vector, in which I've shown the MCS region, which is multiple cloning site. So we have like multiple restriction enzyme cutting sites. We are going to choose any two enzyme and look for these enzymes in the sequence which we have retrieved from NCBI. So supposedly, if we have to clone that sequence between two restriction enzyme cutting sites, we have to make sure that the sequence we are going to clone doesn't have that restriction enzyme cutting sites. Otherwise, what will happen is if the enzyme cutting site is there, then that enzyme will cut the sequence in between which we want to clone. And the cloning will not happen properly because we have already cut the sequence and we won't get the proper sequence which we want to clone. To design a primer to clone that sequence, suppose your HIP1 binding site is some 7 to 8 base pair long. So what you have to do, design a primer. To design the forward primer, you have to take a region 100 to 200 to 150 base pair upstream to that binding sequence. And to make a reverse primer, you have to take 100 to 150 downstream region to that binding site. So, but I have to make sure that if two enzymes which I'm taking for cutting the vector, that those two restriction enzyme sites should not be present in the complete. Supposedly, it makes 300 or 350 base pair long sequence. Then those two cutting sites should not be present in between. So supposedly, I have put that sequence in the software called Snapgene, and this will show the cutting sites of different enzymes. So I have to make sure that those two enzymes which I have chosen should not be present in the cloning region. So I decided to clone my sequence in the site between SAC1 and XMA1. So I have made sure the region I've selected for the forward primer and the reverse primer, the in between sequence region doesn't have a restriction enzyme site of SAC1 and XMA1. So if we look in the snap gene and the sequence in between and the restriction enzyme cutting site in between, none of it has XMA and SAC1 restriction enzyme cutting site. So to clone any sequence into vector, you have to make sure you'll cut the vector and the sequence by using the same pair of restriction enzymes. As I've told you that I'll be cutting the vector with SAC1 and XMA1. So I'll be using the restriction enzyme sequence of SAC1 and XMA1 in the forward and reverse primer respectively. So to make a complete forward primer, you'll be using the forward primer sequence which you have taken from the sequence, and then you'll be putting restriction enzyme upstream to it. And the same case in the reverse primer, you'll take the reverse primer from the sequence, and then you'll be adding restriction enzyme sequence upstream to it. And for both a forward and reverse primer, you'll be using 3 to 5 base pair long overhangs. The length of the overhangs depend upon the restriction enzyme. To know the length of the overhang you should be using, you can check NAB site and you can randomly type your enzyme name, and if the result will be like this. And supposedly I have entered a random enzyme here, and it show good result with 3 base pair, 4 base pair, 5 base pair. So you can add 3 base pair length of overhang. So the complete primer will contain your forward primer sequence. Upstream to it would be your restriction enzyme sequence, and upstream to it would be overhang. And similarly, it goes for reverse primer. It will be having your reverse primer. Upstream to it will be your restriction enzyme cutting site, and upstream to it would be overhang for that particular enzyme. That's it. So if anybody has any question related to the presentation today, they can ask me.
Ma'am, could you just tell us that you initially said that we need to check, like we have to choose any two restriction enzymes that are present in the MCS of the vector, and where all should they not be present in the entire 1,000 base pair region, or just the region that we that would be spanned by the primer and the like the amplicon?
Just the region where you want to just the region you want to clone. Suppose you are cloning 300 base pair region right now, so you will be seeing only that 300 sequence ki whether enzymes like those enzymes can cut there or not. If not, if the enzyme cutting site is not present, you can choose those two enzymes.
Okay, so it is okay if it's like present 500 base pair downstream
Yeah,
of
yeah,
the
surely.
binding.
Yeah, yeah, it
Okay,
won't give any problem to your
okay,
clone then.
okay, all right.
So anyone else? So if no one has any question, then I'll be concluding the presentation now.
Ma'am, is it all for today?
Yeah, it's all for today.
Ma'am, another question.
Right.
So, like, okay, so this, like, I could not understand if it is to be done individually for each transcription factor that we are interested in, or can we do it for all? Like, that portion was not very clear.
Like it depends on your experimental point of view. Supposedly, you are checking for some transcription factor binding site to your gene of interest. And suppose you are interested in only one transcription factor, so you will be cloning that particular region where your transcription factor is binding. But supposedly your experiment is like this ki you don't know which transcription factor can bind to your particular gene. So first of all you will be checking like which all are the transcription factors which can bind, right?
Right.
And then you can choose few among them you are interested to work upon, and then go like singly whether
Okay.
one particular transcription factor is binding to this region, then another one is to this region, and clone like individual section of it.
Okay. Okay. All right. Thank you.
So if there is no other question, I'll be leaving the presentation. Okay.